I guess I'll start with a introduction of who I am. So I'm a machine learning engineer at Booz Allen Hamilton, and I personally have been working in the medical imaging space for roughly the last 10 years or so. Come initially from an academic background, and then joined Booz Allen a few years ago. On the project that I'll be speaking to today, this is a demo that we at Booz Allen, with our CV stack, um, have pulled together to showcase some of our capabilities in the medical imaging space and ability to leverage AI to improve existing clinical workflows. So we'll specifically be focusing on radiology and AI research. But this is, again, largely supported by our computer vision AI stack, as well as collaboration with Voxil, who is a great partner of ours, offering a user-friendly interface and some great data management software, which we were able to integrate with across a number of use cases with our partnership extending into multiple domains. So outside of just healthcare and medical imaging. But today we'll be focusing on the medical imaging piece of that. So just a quick bit of background on, or I guess a broad level overview of what I'll be discussing today. So this is the existing problem that we're honing in on here is building a cohort for large scale medical imaging research projects. So these are generally projects that are going to be IRB approved, involve large teams. There's going to be a combination of AI researchers, developers, as well as large support from clinicians. So this is usually going to require a lot of radiologists on board, and maybe other, cardiologists or other specialties as well, depending on the specific task at hand. The chief problem here that we run into is that engineers in this space, they it ends up taking a huge amount of effort to actually compile these data sets. So a big part of that problem is IRB approval and gaining access to different PAC systems at different hospitals, and figuring out the way to actually integrate with the data and collect that. That's not a problem we're addressing here directly. That's kind of a hospital specific problem, which is going to be a unique solution each time. We're focusing on the second huge obstacle to this data collection, data cleaning process. And that's once you have access and approval to reach into these different data systems at hospitals, how to aggregate that data, how to clean that data, and how to make it AI ready. So the problems in the current workflow, which prevent it from being AI ready once you have that access. This includes requirements for identifying the appropriate imaging. So making sure if you're looking for like a CT study that you're able to identify the exact correct CT protocol, as well as making sure that you have the correct patients identified. So most of these research studies are going to involve specific selection criteria. So I'm looking for patients within a specific age range. I want a balance of patients that have the disease that I'm investigating, with specific representation of that disease, as well as uh, a matching cohort of patients who are going to be that control group who don't have that disease but have similar characteristics. And finally, there's the quality control element. So medical imaging is generally pretty good quality. There's going to be some variation in that, which is hard to avoid. I can show some examples of that. But that'll include imaging artifacts, which occur like when a patient is breathing, which creates motion and kind of blurs and obscures the image, as well as metal artifact, which in some modalities, when you image it, can create this bright kind of starring effect, which makes it hard to quantify anything else in that image. So this is right now a very manual problem. So to do the patient selection, you have to have clinicians come in and investigate electronic health records for those patients, review radiology reports, try to determine if those patients meet your specific criteria. And then determining the imaging itself, if that's going to be appropriate, that runs into a lot of data harmonization issues. So if you're looking at across multiple sites for a multi-site study, then you run into issues with different conventions at different hospitals and trying to make sure that you map those to one kind of standardized definition. And I'll explain that more in detail in a minute. And then the QC parts. So this each image volume contains a lot of 2D slices, and this can range depending on the type of imaging from 20 or 30 2D slices that you would have to scroll through to potentially hundreds. So things like cardiac imaging, you want a very kind of smaller imaging scope, so you end up with and very high resolution in all dimensions, which leads to a very high number of images. So you end up with 500 or more for imaging of cardiac phases. So if you're trying to identify these imaging artifacts, you have to scroll through each of those images manually to ensure that there's not some kind of metal or motion artifact present there. So the solution that we're proposing here is leveraging AI essentially, and then Voxil 51 UI to automate most of this process. So we're trying to do everything we can behind the scenes before users, researchers, radiologists start this data collection, data cleaning process. So we're automating each of these parts. The quality control, we're automating the not the data selection specifically, but extraction of features that are going to be relevant from the imaging data and the patient history data to enable within the Voxil 51 platform users to quickly set filters and go from a very large kind of messy imaging pool down to a very small specific subset, which they're going to be able to use for their study. So taking this process, which could take weeks or months, and cutting that down into minutes. And so this is going to save a significant amount of time, and that both moves the study further along as well as saves time for your researchers, allowing them to focus more in depth on things like model optimization and interpretation of results. And then arguably more importantly, it saves a lot of the radiologists and other clinicians time who are involved in the study, allowing them to focus on the their standard clinical duties. So you're not taking away time from patient care, and then you're also allowing them to spend time on this study, focusing more on higher level decision making, also analysis and kind of interpretation of the results after the AI has run its course. So I'll just give a little more detail on each of these challenges. So we've broken this up into three specific challenges and have developed three solutions accordingly. So the first one we point at, this is that identification of the appropriate imaging. So generally when we're trying to find the right imaging, there's two specific DICOM tags. So DICOM is an imaging, the file type for medical imaging data. There's a few others, but it's predominantly DICOM. And there's study description and then series description are two tags embedded within that imaging data, which are generally what you're going to look at when you try to ensure that the imaging that you're looking for is going to be the right modality, so CT, MRI, ultrasound, as well as have the right specific settings. So with medical imaging, there's going to be a variety of settings that depending on what the radiologist or clinician wants to highlight and be able to visualize more. So you can increase contrast if you want to improve the ability to see specific vasculature or specific lesions, and the amount which you administer that contrast, the timing, again, the modality, and then as well as features like radiation dose are all widely variable and specific to widely variable in the sense of it depends what the clinician is looking for. And the naming conventions for these are essentially all over the place. So you may have a single imaging protocol, which a physician or radiologist uses to look at, let's say, a liver tumor. which has a very kind of standardized amount of, standard modalities. So maybe it's CT, it's contrast enhanced, and your radiation dose and other features are all going to be very aligned. But when you start looking across multiple sites, the way that they describe this in the imaging metadata is going to be all over the place. And this can occur within a single hospital as well. So even if you're focused on a single site and not a larger collaborative multi-site study, you can have uh, image descriptions that are input manually. So uh, these will be generally this metadata is going to be a combination of values that are provided by the imaging device itself, as well as manually input by the ordering physician or radiologist tech. So with those kind of manual touch points, you end up with a lot of variability in naming for the same protocol due to typos, due to preference, due to just different ways people abbreviate. And so you end up with dozens of descriptions that are not identical, but refer to an identical protocol. So this makes it really hard when you're trying to pull together a study with a bunch of different imaging data, and you have a very specific imaging protocol in mind. The current way around this is you look at all the imaging data that's available, you look at all the descriptions of imaging protocols, and then you sit down with a radiologist and you say, this is what we're looking for. What from this list is going to include that image protocol? And it's just a manual process where they have to go line by line and say, and we have some examples here on the left-hand side of the screen. So they may say, thorax width is an example of, or is the identical to these others. So these four are all real descriptions of an identical imaging protocol from the de-identified data that we used for this demo. So, and this is just a kind of a short sampling of it. So again, there could be dozens of different descriptions. So that's the initial challenge. Second challenge here is focusing on the patient selection process. And that generally entails finding patients that are, within a specific age range. You may be looking at specific demographic or socioeconomic factors to try to ensure that you're generating appropriate bias metrics or uh, just building the right distribution for your data set. And right now, the the way to do this is to have clinicians manually go in for each patient, through their EHR system, through their PACS system, look at radiology reports, look at those patients' medical records, and manually review each of those and try to determine has this patient, if you're doing let's say, liver tumor study to stick with the example, does this patient have a history of liver cancer? Yes, no. And then can I have details about that liver tumor? So this is, as you can imagine, a very tedious process. And again, it requires that expertise of the clinician. So you're pulling them away from clinical duties and more kind of important challenges to focus on this tedious, monotonous task. Finally, we have the third challenge, which is going to be that imaging quality control. So this on the screen here, I have an example of metal artifact. So I mentioned there's generally two kinds of imaging artifact that you hit in medical imaging. One is going to be motion artifact. So if you're imaging the heart and the heart is beating, that motion creates kind of a blurring. There's ways around that, but that's a common issue you see, as well as breathing will create motion artifact. So a lot of times if you're getting an MRI or a CT, they'll ask you to hold your breath for a certain amount of time to reduce the effect of that motion on the image quality. And then this metal artifact. So if you have screws from like a prior surgery, or you have some metal implants, with certain modalities, when you do imaging, it creates this very star-like effect, which you can see on the screen. And this one isn't too severe, but you can see how it might obscure part of the image that's important for you. So if you're building like a liver segmentation model and you have this large kind of starring effect, it makes it very hard for the model to identify uh, what type of patterns to learn. So it's that garbage in, garbage out methodology. And that there is a push, to develop generalizable models, but there's a different focus depending on type of project. So when you're looking at a project that's more focused on making a clinical finding, you may want to focus initially on a specific imaging protocol. So narrow your focus to that, a specific subset of patients, and then high quality imaging, where you're not going to run into issues that are due to the image quality and not the underlying clinical patterns. And so this is a method currently very manually intensive to identify that. And again, this requires kind of scrolling through very slowly, investigating each 2D slice, which could potentially be hundreds. And that also includes a very lengthy loading process. So depending on the computer and the system that you're with, but loading each individual scan and then scrolling through that manually. So zooming in on the first challenge here, this is the ensuring that the different descriptions we have from within a hospital or from different hospitals are standardized. So we're working kind of retroactively to make a standard imaging description, which we can quickly reference without having that sit down with the radiologist to determine what's appropriate. So for this, we leveraged BiomedCLIP. That's a clip model that was fine-tuned on, I don't have the numbers in front of me, but I think over 14 million different image text pairs or image caption pairs from PubMed. So this is going to be very clinically focused data. So clip model fine-tuned on a huge amount of medical data. It's performed very well across different medical imaging domains, so radiology, histology, different molecular fields. And so we leveraged it here to try to perform this classification task, essentially. So instead of looking at the provided study description alone, we took the image data, so the pixel values from the image volumes, and then we took metadata. So again, that's going to include a lot of like dosing and other information. It can range from 60 to hundreds or sometimes even over 1,000 different types of values. And we fed those in together into the BiomedCLIP model. So it takes those different data types, the image and then the text values, and then it puts them into an embedding space. And from there, we used an XGBoost model as a classifier. And I'll mention our labels came from, we did have to do an initial sit down to generate the labels with a radiologist expert who was able to tell us that for each image modality or each image volume, we looked at what the standardized description for that would be and what standardized descriptions would be helpful and interpretable by a clinician. So essentially, this model reads in the imaging, the image metadata. It looks at that combination of features and is able to classify this as one of from a list of standardized descriptions. And that takes these, dozens of different protocols and makes it one easy to ingest protocol, easy to filter, easy to search for. The addressing the second challenge here. So this is selection of appropriate patients. For this, we leveraged RadLLama or RadLLama 2. I think there's a more recent version of this that has come out, but for when we generated this, we used RadLLama 2, which was a 7 billion parameter model. So Llama transformer model coming from Facebook or Meta. And this was fine-tuned by the Stanford AI Medical Imaging Group using millions of radiology reports and other textual clinical data, and has performed very well on a variety of radiology tasks, specifically radiology reports. So our data set that we used had a number of radiology reports available. So we threw this model at them and were able to extract a bunch of clinically relevant features. So looking at these unstructured free text radiology reports, ingesting that and then extracting the features that we think might be relevant for identifying whether a patient's appropriate for a cohort. And then we take that and structure it and embed it into the DICOM metadata, again, to make it something that's searchable, that's filterable. So this could be something like this patient, did this patient have cancer? Yes, no. Do they have liver cancer? Yes, no. What's the size of the tumor? What is the precise location? What's the volume? what are specific characteristics of it. For the third part of the solution, we look at the quality control piece. So for this, again, there's the image artifacts. The two primary are metal artifact and then motion artifact. So we started actually with an initial segmentation of the full volumes. So we used Total Segmentator, which is based on I think it's no new UNet on the back end, which is a UNet-based framework that's been specifically developed for medical imaging. So it can quickly and accurately segment different anatomy, so organs as well as musculature and skeletal pieces as well. And so we ran that, and then we did a detection of imaging artifacts, and then looked for correlation between those. So this allowed us to say, hey, there's metal artifact in this scan, but specifically this metal artifact is occurring in the liver. And this enables clinicians or researchers, when they're trying to filter through their data, to isolate the imaging artifact to a specific area and determine if that's going to be relevant for them or not. So you can imagine if you're doing a study on lung cancer and there's a motion artifact or I guess probably metal artifact more likely in the liver or pancreas or somewhere that doesn't obscure anything in the lung part of the scan, that's probably not as relevant to you. But if you're doing a pancreas study and you have metal artifact in the pancreas, that might be something that disqualifies that imaging from your study. So this combined approach allows us to both detect those anomalies and then isolate them to specific regions. And so we took the output from these three models, or these three components of our pipeline, the patient selection, the image protocol classification, and the quality control element with that kind of isolation to specific anatomy. And we embedded that into DICOM metadata. And then at that point, you have all the data available to kind of quickly, efficiently filter through a patient cohort, set filters like I'm looking for patients between 25 and 45. I only want patients that have had contrast enhanced CT of the chest. And I specifically interested in patients with a history of liver cancer with at least 2 tumors of this specific volume. The missing piece there is there's no ability there to actually leverage those filterable items, and so that's where Voxil 51 comes into play. So Voxil's 51 UI is a quick, efficient way to be able to leverage a lot of these metadata values that we've supplied. So you can see in the example here, you're able to load in huge amounts of data. So you can start with your very large kind of messy protocol, or not protocol, your very large data pool. You can search and kind of explore specific patients. You're able to directly load in the segmentation model output from the QC part of our pipeline. And you can efficiently scroll through and kind of investigate different areas in that data exploration process. You can hover over some of the segmented anatomy and it'll show you specific metadata about that. So things like what's the volume of this particular region. And then you can also explore the embeddings. So here, this is going to show the embeddings of our image protocol classification model. And you have the ability to color code this based on the labels. So we can break this down into the labels for each of our standardized image descriptions. And it's as interactive as well. So you can click on each of these specific labels and pull up all the imaging that would fall under that label and investigate the model's classifications, ensure that that's aligned with what you might expect. And let's see. There's also an interactive dashboard available. And so this is very customizable. You can use it to explore different patient features that are present in the image metadata, the imaging itself, as well as other features that you load in. And this can allow you, once you've gone through that process of setting those specific filters to define your imaging type, to define your selection criteria, to define your quality control thresholds. Once you've narrowed down from, thousands to the hundred or so appropriate patients for your study, you can use this to explore features within that. You can ensure that you have the right distribution of different demographic features or specific imaging characteristics that you need for a robust, significant interpretation of your study. And that brings us to the end. So yeah, would like to thank Steve Caston, who's the radiologist subject matter expert who supported us with a lot of um, the labeling, as well as just insight into what clinicians would find useful, interpretation of different imaging features and identifying yeah, anything that might be relevant for us. Farhad, Mukund, Roger, Sarah, all across the Booz Allen side, supported a lot of the development on this. And then big thanks to the Voxil 51 team, who worked closely with us throughout. And I'll stop there for questions.
Yeah, fantastic. So I'll give you a few questions. We'll kind of start at the questions that came towards the end. How many records in your labeled data before you felt comfortable with the predicted results?
I guess which part of the pipeline would that question be referring?
I would assume probably maybe take it from the training piece of it.
Okay, but I mean it would it's going to vary depending on part of the pipeline. And I'll also meant so the models, most of what we use are these large kind of foundational pre-trained models. So from a training perspective, we did train the classifier head, the XGBoost, on the image protocol classification pipeline. So that kind of solution two, I think, for our pipeline. Um, how many like labeled pairs do we ingest before or I guess I'm not totally following the question. Like how much training data do we need before we're confident in predictions or
Yeah, CC, why don't you follow up on that question with a little bit more specificity? And if we have time, we can circle back on it. I think you may have covered this in the challenge section, but early in the overview section, it was asked, would metadata include instrumentation marks or overlay on a rendered image like a CT or MRI?
I guess sorry, I'm struggling again. The instrumentation marks, is that
I would think those are the metal objects, perhaps, that you were talking about.
oh, okay. And sorry, could you repeat the question? Does
Yeah. Would metadata include instrumentation marks? Let's just go with that's the metal objects, or
okay.
overlay on a rendered image like a CT or an MRI.
Okay. I'm gonna take those separately. So for the would metadata mention that there's metal artifact? Generally, no. That's not something that would be automatically input. Like the imaging vendor or the imaging device would not automatically detect or put that into the metadata. It's possible that there are these kind of free text entry points for, again, potentially over 1,000 different tags, which can be provided by a rad tech or the ordering physician. It's possible that like a radiologist technician, when they're doing the imaging, would note that if they're trying to image the liver specifically and there's metal artifact in the liver, and that's causing them an issue such that they need to redo that imaging a few times and kind of tweak settings, they might call that out specifically, and that would be embedded in the data, but generally no would be the answer. And then I took the second part of that to mean annotations specifically. So things like um, the segmentation masks that we showed, or bounding boxes or other annotations. There is a standard way to embed that into DICOM metadata. So that generally that's done later in the process. Like you wouldn't do that type of annotation unless you had a study which specifically required it, or if you were trying to do sizing for a specific device that you might want to implant, like you might manually trace different contours in the heart if you want to do an implanted cardiac device, and that could be stored to within the metadata, but I think that's infrequent and requires a specific pipeline where you would need to do that.